Now let's look at the other half of the lecture and learn about immunoassays, nucleic acid tests, and various laboratory diagnostic tests for viral pathogens such as plaque assays and plaque reduction neutralization tests and hemagglutinin inhibition tests. There are two major types of diagnostic laboratory tests. The first are immunoassays, which are based on the antibody-antigen interactions that were discussed last week. And the second group are the nucleic acid tests, which use genetic variation to identify and differentiate various pathogens. Immunoassays can be designed to detect either antibodies or antigens. For antibody detection, immune responses to pathogens are determined, and diagnostic tests ascertain the increase in antibody titers or levels during and after infection. This is usually IgG and IgM levels in blood specimens. For antigen detection, the presence or absence of microbial pathogens are determined by testing for certain antigens that are definitive markers for the disease. The clinical specimens submitted should be the most appropriate ones for the particular disease. These are usually specimens which have the highest likelihood of pathogens being present. For example, when testing for gastrointestinal diseases, stool or fecal specimens are usually the best clinical specimens to collect, and blood for blood-borne pathogen. Immunoassays have several major advantages. They have good specificity and sensitivity. This means that they are designed specially so that the likelihood of getting false positives or false negatives is as low as possible. This is important because the diagnostic test should not be positive when the case is not infected and should not be negative when the case is actually infected, as this could lead to potential therapy and psychological problems in the case of false positives, and also more serious disease transmission and prognosis issues in the case of false negatives. Immunoassays are usually used when microbial pathogens cannot be cultured or isolated easily from clinical specimens. This mainly applies to the more fastidious bacterial pathogens and also a large number of viruses. They are also used when antibody responses are a good marker for infection and exposure. Lastly, they are used to type microbes that look identical under microscopy but may have different pathogenicity based on their serotypes. This is the same slide that was presented last week. It also applies to immunoassays, as the time the serum specimens are collected is important. Since IgM is produced early during the first infection, high levels of IgM are good markers for a current or very recent infection, as IgM levels then drop after 2 weeks. Elevated IgG levels, however, are usually good markers of historical exposure, either from previous infections or through vaccination. High levels of IgG also indicate that there is likely some protective immunity present, and this is above a certain threshold level. The individual will not require a booster or a fresh dose of vaccine. It is sometimes important to determine this after accidental occupational exposures in the workplace to certain pathogens, such as the hepatitis B virus, and to decide on the type of post-exposure prophylaxis required. Let's look at immunoassays at the molecular level. One of the most common assays are ELISAs, or enzyme-linked immunosorbent assays. These involve using plastic plates with many different wells that are called microtiter plates. The plastic surfaces in the wells are specially coated to make them sticky to antigens and antibodies and other biomolecules. So if we're detecting antibodies, the first step is to coat the surfaces of the wells with antigen from the pathogen that needs to be tested. I have represented them as pink triangles. It is then essential in step 2 to block any remaining uncoated sites on the surface. These are represented as black discs. False positive results can be obtained if these non-specific free binding sites are present. So a protein solution such as skim milk or bovine serum albumin is usually placed in the wells and left overnight to bind to the remaining surface. After the blocking reagent is removed, the plates are ready to be used. In step 3, wells are then incubated with test serum, usually for an hour at 37 degrees Celsius or body temperature, to allow any antibodies that might be present to bind to the antigens. They're then washed thoroughly to remove any unbound antibodies. These are antibodies that bind to other antigens and are therefore not relevant to the test. They may also interfere with the test. After washing in step 4, the second antibody, which is usually either anti-human IgG or anti-human IgM, is added and incubated for another hour at 37 degrees Celsius before another wash step is carried out to remove any unbound antibodies. These second antibodies are conjugated with enzyme that will result in a visible color change if the specific antibodies are present. In my animated diagram, we're looking for a green color as the positive result. This is a picture of a lab technician performing an ELISA. She is using a multi-channel pipette to dispense reagents simultaneously into the wells of a microtiter plate, which usually has 96 wells. I'm also going to show you 2 videos on different plate washing techniques. The wash steps are important because any leftover antibodies will also cause non-specific reactions that will yield false positive results. So it is always necessary to include negative controls to determine if the blocking and wash steps are carried out properly. If the negative control wells turn positive, then the assay is flawed and needs to be repeated as the results are unreliable. After all the incubation and wash steps, this is the anticipated result. You can see some wells which are green. There are 4 sets of 4 wells in the center of the plate. The 4 wells are replicate wells using the same serum specimen. And the 4 sets are 4 different serum specimens, which are indicated in the wells that have different color borders around them. The green wells on the bottom row are the positive controls, which are added to determine whether the test has worked. In this case, you can see that the test has worked properly. If no color change is present in the positive control wells, it means that the test did not work and false negative results were obtained. Once again, the test needs to be repeated as the results are unreliable. The intensity of the color can be measured and the antibody titer and level determined. I've drawn a diagram of a sample ELISA microtiter plate to explain the results in greater detail. On this plate, serum specimens were added to 3 replicate or triplicate wells, vertically in columns rather than horizontally in rows, as in the plate on the last slide. You can see these triplicate wells in various shades of green. In the top 3 rows, that's rows A to C, acute serum was added using various dilutions. These are doubling dilutions that range from neat undiluted to 1 in 2, then 1 in 4, to the final and 10th doubling dilution of 1 in 1024. In the next 3 rows, D to F, convalescent serum from the same case was also added in various dilutions. The convalescent serum is usually collected when the patient is recovering. The antibody titer is the last dilution that produces a positive result. In the case of the acute serum specimen, this is found in wells with the 1 in 4 dilution. For the convalescent serum specimens, the antibody titer is 1 in 128, as the green color reaction is only present at that dilution. It is absent in the wells with the next doubling dilution, which is 1 in 256. This means that for the case, the antibody levels increased by 32 times during their recovery period, indicating that they've mounted a good immune response to the pathogen and that these results are positive confirmation of their exposure. There are several notifiable diseases in the National Surveillance Program that use increased serum antibody titers for lab confirmation. The criterion is a 4-fold or greater increase in antibody titers, so for the example in the last slide, the case will be considered to be a lab-confirmed positive case. These diseases include syphilis, brucellosis, plague, and tularemia, as well as a large number of viral diseases such as HIV, flu, hepatitis A, B, and C. Other lab tests that use antibodies include agglutination tests. Unlike ELISAs, these are rapid tests. They provide distinct and clear results and usually involve the observation of white latex agglutination against a black background. They're not as sensitive as other immunoassays, and false negative results may sometimes be obtained. Examples of agglutination tests are the identification of the Streptococcus lanceolus groups. The picture I've drawn shows IgG antibodies conjugated to latex beads that are binding to the antigenic parts of the pathogen. which I've represented as neon pink circles. You can see the formation of an immunocomplex. To carry out the agglutination test, small suspensions of bacterial cultures are placed on the test card, either on different sides or in circles. The antibody is then added and mixed with the bacterial culture by tilting the card back and forth for a minute. If clumping is observed, a positive reaction is obtained. This can be seen in the upper side of the card in the picture, whereas on the lower side of the card, the suspension is smooth and there are no clumps, indicating that agglutination is absent and a negative result is obtained. In the last slide, only one antibody was used, but on this slide, I have added a typical latex agglutination test for serotyping Streptococcus and determining the Lancefield groups. You can see here that 6 different antibodies were used and placed in different circles on the card. Each antibody binds specifically to Streptococcus bacteria that belong to the 6 different Lancefield groups. Which group do you think this bacteria belongs to? Well, you can see clearly that clumping is present in circle number 3, which has the Lancefield group C antibody added to it. So the Streptococcus bacteria in this case belongs to group C. Before we carry on to the last type of immunoassay, I thought I should introduce you to the different types of antibodies that are used in immunoassays because you may come across them in your professional practice. The antibodies that are present in serum specimens vary from person to person depending on their exposure histories. All individuals will have a selection of different antibodies that are able to bind to different epitopes on different antigens from different pathogens. You can see from my crude diagram, one antigen alone can have several epitopes and several different antibodies that bind specifically to each epitope. These antibodies, which are usually found in serum or antiserum, if the antibodies are raised by hyperimmunization of animals, are referred to as polyclonal antibodies. There is another type of antibody that is produced artificially in either mice or tissue cultures or by genetic engineering. These antibodies are specially designed to bind to only one epitope of one antigen of a single pathogen, so they are extremely specific and are called monoclonal antibodies. As the name indicates, they are produced by clones of a single antibody-producing B cell. In order to produce large quantities of this type of antibody, a novel method was developed, in which mice were inoculated with the antigen of interest, then their spleen cells were harvested. These are the light blue cells in the diagram. They were then mixed with the B-cell myeloma or cancer cells, the orange cells. A special reagent was then used that enabled the 2 different types of cells to fuse together, resulting in a hybrid type of cell called hybridoma cells. In the diagram, I've colored them light green with orange cell membranes. These unique hybridoma cells were able to produce antibodies to the antigen of interest and could be grown in culture since they had the immortality of the myeloma cells. In order to obtain only one type of antibody that binds specifically to only one epitope, the hybridoma cells were cloned. One method of doing this was using limited dilution, where a suspension of hybridoma cells were diluted to the extent that only one cell would be present in one well of a microtiter plate. This one cell then divided to produce 2 identical daughter clone cells. Further propagation of these clones enabled large amounts of antibodies to be produced. All the antibodies were identical and bound only one type of epitope. These identical antibodies are called monoclonal antibodies. Because they're so specific, they're used in many different types of immunoassays, including ELISAs, agglutination, and immunofluorescence assays, which we'll look at next. Immunofluorescence assays, or IFA, are very similar to ELISAs. However, instead of using microtiter plates, microscope slides are used in order to be able to view the fluorescence using ultraviolet light microscopy. If you remember some of the material in ENH 222, whenever you place bacteria or any other kind of microbe on a slide, you need to fix it so that it doesn't fall off. Do you remember doing this before you performed your gram stain? Can you remember how you fixed your specimen? If you remember the Bunsen flame, you are correct. For IFA slides though, heat is not usually used to fix the specimens because this can denature the antigens and make them unable to react with antibodies. So a variety of chemicals can be used instead. These can be alcohols, acetone, formaldehyde, or glutaraldehyde. Once the smear is fixed, it is incubated with the first antibody. In direct immunofluorescence assays, this first antibody or primary antibody is usually a monoclonal antibody, or mAb for short. Using this mAb enables the specific antigen to be detected and identified. The monoclonal antibody is labeled with a fluorescent dye, so if the antigen is present and the antibody binds to it, it can be easily seen under ultraviolet light illumination. The method is usually very sensitive, as bright green glowing fluorescent microbes can usually be seen clearly against a dark black background, especially if the washes are done thoroughly. Indirect immunofluorescence assays are even more similar to ELISAs, as there is an added step with a second antibody. Usually, polyclonal immune serum is used for detecting antibodies to specific antigens. A monoclonal antibody may also be used if specific antigens are being detected. Indirect immunofluorescence assays are more versatile than the direct ones because the second antibody is the one that is labeled with the fluorescent dye. This means that this type of test can be easily adapted for detecting different antigens or antibodies, and it is not necessary to label the primary or first antibody, which can be a difficult chemical procedure to carry out in a microbiology lab. This is because secondary antibodies are usually purchased from specialized commercial suppliers who are able to produce large amounts of the antiserum in animals, whereas the primary antibodies are usually developed in microbiology or immunology research labs. This is what an immunofluorescence stain slide looks like. The microbes on the left slide are the gametocytes or sexual stages of the malaria parasite. In this indirect immunofluorescence assay, I used sera from school children in Papua New Guinea as the primary antibody. Then I added goat anti-human antiserum labeled with a green fluorescent dye. The results show that school children in Papua New Guinea had antibodies to Plasmodium falciparum gametocytes. These are the stages that develop in mosquitoes to produce new generations of parasites. Although this is not a surprising result since malaria transmission is endemic in Papua New Guinea, what was surprising was that the children were not sick and were running around in school playgrounds. It is very possible that the antibodies they had protected them from severe disease. On the right are similar gametocytes that were stained with a histological stain used in all malaria labs to show you what they look like normally against a background of donut-shaped red blood cells. Most nucleic acid tests involve the detection of either DNA or RNA of microbial pathogens. This is carried out using hybridization or binding of oligonucleotide probes that have complementary sequences to the target gene sequence in the pathogen of interest. If you look at the sample sequence below, you can see that the oligonucleotide probe is only 15 bases long, and it binds to the complementary target sequence. Like the immunofluorescent assay antibodies, the probes are usually labeled with fluorescent dyes. Probe hybridization is useful for the detection of specific virulence genes and microbial pathogens that are difficult to culture. So I've provided an example of how microbes which look identical but have different genotypes and DNA sequences can be typed. You can see two different DNA sequences. The first, G1 genotype, belongs to a non-pathogenic strain, which has a different sequence at the target site of the probe to the G2 pathogenic strain. The probe has a sequence which matches the target sequence of the G2 genotype completely, so binding occurs and a fluorescence signal is produced, whereas no fluorescence is observed for the non-pathogenic strain. A similar kind of hybridization is also used in the polymerase chain reaction, or PCR, which is now used in many diagnostic nucleic acid tests. Shorter oligonucleotides are used. Due to their different function in PCR, these short oligonucleotides are called primers instead of probes. There are two of them, the forward primer and the reverse primer, and they flank the 5' end and the 3' end of the target gene locus. To set up the master mix for PCR, you need to add the pair of primers and a heat-stable polymerase, such as TAG. This DNA polymerase is derived from a thermophilic bacteria called Thermus aquaticus, which was first discovered in the hot springs of Yellowstone National Park. The nucleotides, which are the building blocks of DNA, also need to be added to buffers and salts. Once this master mix is prepared, the clinical or test specimen can be added. This contains the template DNA. The clinical specimen needs to be processed and the DNA extracted before it can be used in PCR. The tubes are then placed in a machine called a thermal cycler that is able to change the temperature of the heating block in program cycles. Each cycle includes 3 steps, carried out at different temperatures. I will go over the steps one by one in the next 3 slides. The denaturing step is carried out at high heat to separate double-stranded DNA. In this denaturing step, the temperature is set at 94 degrees Celsius for 1 minute so that the double-stranded DNA can be separated into single strands. The primers, nucleotides, and DNA polymerase have previously been added in the master mix. The annealing step is then carried out at a lower temperature to allow the primers to bind or hybridize to the DNA sequences that flank the target sequence. The temperature is usually lowered to around 60 degrees for 1 minute in the annealing step, in which the forward and reverse primers hybridize to the short stretches of DNA that flank the gene locus that is being amplified. After the annealing step, the temperature is increased to 72 degrees for 1 minute in the extension step, during which the DNA polymerase synthesizes the new strands of PCR product. The DNA polymerase extends the new strands of DNA by incorporating the nucleotides from the master mix using the original strands as templates. 2 copies of the target DNA are produced after the first cycle. This cycle is repeated usually 40 times till enough product is obtained for analysis. As the steps are repeated or cycled, more copies of the target DNA are produced till after 40 cycles the amount is increased exponentially. This is why it is called a chain reaction. The PCR products are then analyzed using agarose gel electrophoresis. This enables the size of the product to be determined. This is done by running a set of molecular weight markers alongside. Because DNA is not visible, it is stained with fluorescent dyes. In the old days, ethidium bromide was used, but this chemical is highly toxic and a mutagen, so molecular biology labs now use less toxic fluorescent stains, such as cyber-based dyes. After staining the gel, the DNA bands are visualized using a UV transilluminator and the bands captured by photography. You can see the black and white photo of the gel from the last slide. Most labs now use a digital camera, and the data can be stored and archived as computer image files. I'm not sure what the bands represent because it's from a lab in New Zealand and was posted on Wikimedia with a Creative Commons share-alike license, so I've used it. You can see the DNA ladder in the first lane on the left-hand side of the gel. These are the molecular weight markers, but I don't know their sizes because this wasn't provided, so I've used my own gel to share with you. This is one of the gels from my old research lab at the BC Centre for Disease Control. My research team was investigating a waterborne outbreak of cryptosporidiosis in North Battleford, Saskatchewan. We were invited by the Health Canada epidemiologists to participate as my group was the only one in Canada at the time who had the PCR method up and running. We were sent fecal specimens from outbreak cases and amplified the small subunit ribosomal RNA gene, which is a good genetic marker for cryptosporidium. Based on the molecular weight markers on the last lane on the right-hand side, you can see 2 bright bands of DNA at around 400 base pairs in size in lanes 3 and 4, which were amplified from specimens of 2 different outbreak cases. You can also see the same band in lane 6, which is the positive control, although it's not as bright as we're trying to save the positive control template and was using it sparingly. The negative controls in lanes 1 and 7 were blank, which is great, as it showed that we didn't get any false positive results. We then sequenced the PCR products to determine their DNA sequences, which provided us with information on the course of the outbreak. We determined that the contamination came from human sewage, and confirmed that it was the wastewater treatment plant effluents that contaminated the community drinking water supply. The drinking water treatment plant, which was located downstream of the wastewater treatment plant, had malfunctioned, and there was a breach in the water filtration system after it was shut down for cleaning and put back into service. The city then switched to an alternative groundwater source, and the number of cases dropped. This is a picture of a lab technician setting up a PCR test in a clean room. He is using a biosafety cabinet to protect himself as well as the specimen from contamination. All the pipette tips have filter plugs in them to help reduce cross-contamination between specimens, and at the end of the day, the cabinets are irradiated with UV light to break down any DNA that might be left on surfaces. More and more communicable diseases are being confirmed in the labs using PCR. These are mainly viral diseases due to the difficulty in culturing pathogenic viruses. Currently, for bacterial diseases, pertussis and invasive meningococcal disease can be confirmed using nucleic acid tests. So in summary, the polymerase chain reaction is very sensitive, as it is able to amplify large amounts of DNA from a very small amount of template DNA. It does suffer, however, from contamination problems, and false positive results are a common problem. To prevent contamination, dedicated clean rooms are required for specimen and master mix preparation. Negative controls need to be included in every test run to ensure that no contamination is present and the results are not false positives. Many pathogenic viruses cannot be cultured in cell lines, so their identification has been a challenge. The polymerase chain reaction has enabled the diagnosis of many viruses, as large amounts of DNA can be generated from the small amounts present in a clinical specimen. We've already gone over the fundamentals of PCR, which provides the ability to confirm the diagnosis of many more viral diseases and infections than was possible in the past. And this is fairly recent because it has only been used in routine clinical diagnostic tests in the past 20 years. For RNA viruses, the enzyme reverse transcriptase needs to be used to convert the unstable RNA to stable DNA for further analysis. Hence the term RT-PCR is used instead of just PCR. Real-time PCR or qPCR is a recent modification of the standard PCR technique, enabling rapid diagnostic tests that can be quantitative when using some specialized instrumentation. It is called real-time because you can actually monitor the PCR results while it is happening, so no additional wait times are required for a diagnosis.